Okay, so good afternoon, everyone, and welcome back. And let's try finish our discussion of mitosis today. And if possible, I'll move on to discussing the next kind of cell division, which is meiosis. But we'll see how far we get with this today. Let me just run over the different stages of mitosis. Remember that mitosis is the cell division which results in two identical cells being produced from a parent cell. It's the division which is associated with growth, with development, and also with repair, that sort of process. Mitosis involves diploid cells, that is, cells with their full genetic complement, which divide to produce two identical cells, each again with their full genetic complement. A diploid cell divides in mitosis to produce two diploid cells. You'll see later on when you talk about meiosis why I'm laboring that point to make it strong for you. So let's just have a look at the different stages that we saw last time towards the end of our lecture. And let's look first. The longest stage of the cell's life is interphase. In interphase, this is the normal functioning period of the cell. This is when the cell does whatever the cell is meant to do. But at certain points during interphase, during the synthesis phase of interphase, the DNA is entirely replicated. And in addition, these two little bodies called the centrosomes are also duplicated. And towards the end of interphase, as we approach at the end of the gap 2 stage, when the cell is preparing to enter into mitosis, into cell division, these two centrosomes begin to move apart. This is often the first sign that cell division is impending. They move apart, and you can see here they begin to spin microtubules. They begin to manufacture microtubules. So the very first stage of mitosis itself is called prophase, and we recognize prophase because during prophase the chromosomes begin to condense. They begin to get wound down into tighter and tighter and thicker chromosomes, which become visible. You can see them just appearing here. At the same time, the nuclear membrane disappears. The nuclear membrane breaks down because mitosis cannot take place if the membrane is present, for reasons that will become obvious. If we watch what's happening with the centrosomes, we see that they're actually beginning to move, and they're beginning to move apart. They're going to move apart to opposite poles of the cell. At the same time, they are making these microtubules which link up with one another and also produce this kind of rayed look. This is called the aster. Aster meaning star, because that's effectively what it looks like. And it's the centrosome with the microtubules radiating out from it. These across here form a spindle, and that is referred to as the mitotic spindle. So the next stage, after prophase, is prometaphase or early metaphase. You'll see it referred to as both. The nuclear membrane is gone, it's disappeared. And instead, the chromosomes now, instead of looking like they used to with just being grainy chromatin, they now condense into visible chromosomes here. And this spindle has made its way all the way across, and you'll see that some of the microtubules are actually contacting the chromosome there at the centromere. And they're actually contacting a special apparatus on the centromere called the kinetochore. So we call these the kinetochore microtubules. Here they are here. There's one attached there to this chromosome, one attached there to this chromosome. But also notice that there are other microtubules, and they are contacting one another and overlapping. They have a special function we'll talk about in a minute. So we have non-kinetic core microtubules, and then the kinetic core microtubules contacting the chromosomes there. If you sit and watch, you would see that the chromosomes appear to move up and down these microtubules. They kind of jiggle back and forth. We'll see it in a little video in a minute. They kind of jiggle back and forth and back and forth and back and forth. Towards the end of pro-metaphase, they start to make their way to the central plane of the cell. It's an imaginary plane cutting across the center of that of mitotic spindle. We call it the metaphase plate. And the chromosomes all become oriented on that metaphase plate. They jiggle around until they're exactly in position. Nothing more will happen until they are all positioned exactly on the metaphase plate. And that happens towards the end of metaphase. And immediately it's accomplished, we'll see the chromosomes split apart. And what's important here, look at this chromosome here. What does it consist of? It consists of 2 chromatids, and those chromatids are identical to one another. They were replicated during interphase. The 2 chromatids are identical, and during the next phase of division, anaphase, those 2 identical chromatids split apart. And during anaphase, these chromatids move quite rapidly to the opposite poles of the cell. They're moving on those microtubules. We'll discuss the method that they move by in a second. And once that is accomplished, once they reach the other side, the chromosomes immediately begin to unwind. They begin to adopt their normal construction. The nuclear membrane begins to reform around them, and the nucleus begins to assume its normal functions. As you can see here, this stage here where the nuclei have been split apart from one another like that is called telophase, and it marks the resumption of normal nucleus structure and normal nucleus function. Once telophase is accomplished, you've got the 2 well separated, then the actual cytoplasm of the cell divides, and that is called cytokinesis. Cytokinesis is regarded as an entirely separate process of the cell dividing in 2 to separate 2 new nuclei, one from the other, as you can see here. So if we have a look here on the metaphase, you can see the chromosomes all aligned here on the metaphase plate at the equator of the cell. Here is anaphase. The identical chromatids are being moved apart from one another. And here is telophase. The rest of the chromosomes now starting to unwind, and the nuclear membrane starting to form again. So I just want to mention one thing. During anaphase, the most obvious thing that happens is that we see the chromatids moving apart from one another. But less obvious is the fact that these microtubules here, the non-kinetic core microtubules, they are overlapping one another. And on that overlap there are motor proteins. And the motor proteins force the microtubules to slide against one another and away from one another. And that causes the cell to elongate. There's an actual physical pushing of the cell, of the 2 poles of the cell away from one another, along with the telophase features of the nuclear membrane reforming and the chromosomes unwinding to form the new nuclei. Okay, so remember, there are 2 functional roles for microtubules. One is the movement of the chromatids. The other is actual physical pushing apart, lengthening of the cell to get them even further apart from one another. Okay, so just to mention re-emphasize one thing that I have said to you before. Interphase is the normal functioning period of the cell. And during interphase, all the preparatory steps are taken for cell division to occur eventually. Principally amongst them is growth of the cell, increase in biomass, essentially. But the period of mitosis, the M phase of mitosis, is very, very short, and necessarily so because most gene function has to be silenced during M phase. So none of the normal gene function can be performed, and that's a very dangerous situation. The cell there is depending entirely on materials and enzymes and everything else that it has laid down during interphase, especially during G2, when it really gears up for the cell division. So mitosis is necessarily the shortest phase of all, so that the cell, as much as possible, spends most of its life with all of its genes functional. Okay, let's see if this works. This is animal mitosis. One of them's lost. There he is. As soon as they all lined up, there they go. Okay, so I just want to mention a couple of things about the functioning of the microtubules. First of all, have a look here, and you can see the structure of this centrosome radiating out microtubules in all directions to form this aster around the centrosome itself. But here are the microtubules, which are extending all the way across the cell. We have the non-kinetochore microtubules here overlapping with one another, but it's these ones here that we want to pay particular attention to for the moment. These are the kinetochore microtubules, which attach themselves to the centromere of each chromosome on either side. It is tempting when you look at these to think that the microtubules are actually pulling and pushing like this, but they for the most part, they aren't. There is a little bit of that does occur, it has to be said, but most of the movement of the chromosomes is the result of the activity of this very complex set of proteins, of motor proteins and things which are attached to this complex of proteins here at the kinetochore. Kinetochore means the center of movement. So this is actually what is producing the movement of the chromosomes, of the chromatids, especially apart from one another. So realize that in between here, there's one chromatid on this side and one chromatid on this side. So, and we're going to watch them move apart from one another. What actually happens is that there's a set of motor proteins on the kinetochore which move the chromatid along the microtubule as if it was a railway track. So the motor proteins are actively marching the chromatid along like this. And then the kinetochore also has the ability to break down the microtubule behind it. So as it moves like this, the microtubule breaks down behind the chromatid. This they actually call it the Pac-Man mechanism, just to remind us of the old video game. What's happening is the kinetochore is kind of moving along and chewing its way along the microtubule to walk the chromosome towards the pole. And this was demonstrated in a very clever little experiment. What they did was they looked at cells that were busy dividing, where the spindle was really obvious, and right at the beginning of anaphase, they stained them, and then they bleached part of the spindle with a laser beam. So here's the normal spindle, like this. The chromatids are just beginning to move apart in anaphase. They bleach this part quickly with a laser, and then they watch. At this time, there was an argument. Some people thought that the microtubules were attached to the chromatid, and then they were reeled in by at the spindle pole. They were kind of pulled towards the spindle pole. Other people said no, the chromatids were actually marching along. So if they're being pulled like this, then these marks will move towards the spindle pole, and the chromatid will stay a constant distance from the mark. They didn't. Instead, it was very obvious that the chromatids were this mark stayed more or less stationary, and the chromatid marched towards it and eventually overcame the marks in actual fact. And that was proof that the major movement was induced by something on the chromatid itself. It turns out that there is a little bit of pulling. There are some motor proteins here which do in fact pull as well. But the major movement is like this. Here's the kinetochore here. Here's the chromosome, the chromatid, right? Here's the kinetochore attached at the centromere. And there's motor proteins here which are moving it along the microtubule. And at the same time, it enzymatically digests the microtubule behind. So that's a neat little experiment. Now the cleavage, the cytokinesis of the cells differs a little bit between animals and plants. Remember that plants have a cell wall around the cell, which has to be accounted for. In animal cells, there's no cell wall. The cell can simply divide on its own. And it does, in animal cells, it does so by pinching in. It's called a cleavage furrow. And in actual fact, it's a very complicated action of the cytoskeleton. Actin fibers actually form in a band around the cell and then move against one another to pinch the cell down, they're causing this cleavage furrow and then eventually separating the two cells. In plants, a slightly different process takes place. Here is a transmission electron micrograph of a plant cell. Here are the cell walls on either side. Here's the dividing plant cell in the middle here. The new nucleus on one side, the new nucleus on the other side. But the plant has to lay down a cell wall between the two new cells. And it does so, you can see it being laid down here. It does it along what is called a cell plate. So this is laying down new cell wall materials, cellulose, whatever, etc., to form the cell plate. And then the cell itself undergoes cytokinesis behind that, so that the cell becomes separated, two cells become separated one from the other by the cell wall, and then the membrane is entirely healed to form the two different cells on either side. Yeah, let's have a look at these couple of photomicrographs of plant cell division. Essentially, the division is mitosis is very, very similar in both plants and animal cells. We'll see the same stages exactly. But in plant cells, often they're nice to look at because they're very, very easy to recognize. So here, prophase, in a plant cell. You can see the chromosomes just beginning to condense. Here's pro-metaphase. This is early metaphase. The only thing that you'll notice with plants is the spindle is not as obvious as it is in animals, but it's still there. This is metaphase with them beautifully lined up on the metaphase equator. And here's anaphase and telophase. And again, during telophase, we see here formation of the cell plate that will eventually move right across there. And then the cell membrane just heals up behind the cell plate and that amounts to cytokinesis. Here's a favorite exam question to give, but I don't know that I'll be able to fit this into one of your exams. But have a look at this and see how many different phases of cell division you can identify in this photomicrograph. This is actually onion root, and this is also a favorite exercise in lab, to look at onion root and see how many different phases of mitosis you can see. How about that there? What would you say that is? Yes, probably prophase. What about this here? It's beautiful. You can't mistake that. That's anaphase. What about this here? Perfect. Textbook metaphase. What about this? Just this one here? Maybe there's another one we can use. In any case, yeah, use this one here. Well, that's early telophase. You can see the cell plate being laid down in between, etc., etc. So you can go through these and see if you can identify all the different stages. Unsurprisingly, most of them are in telophase because that's the longest period. And the cells that are undergoing mitosis are doing so very rapidly, so there are a lot fewer of them. Okay, I just want to say a word about what happens in prokaryotes, because prokaryote cell division is not mitosis. It resembles mitosis in that the DNA, the genome of the prokaryote has to be replicated entirely, and then has to be, the cell has to develop so that we split the two copies of the DNA, one from the other. In prokaryotes, there is one very large circular chromosome, and that is replicated. The chromosome is actually attached to the cell membrane at one point called the ORI, O-R-I, ORI-C is the proper name for it. So, and replication begins there, but we end up with two circular chromosomes, and the attachments to the cell membrane are actually moved. They are physically moved apart from one another in a mechanism which we do not know. We don't understand exactly how that takes place. But nonetheless, we end up with two copies, and there are two origins which are attached to different places on the cell membrane. You can see here and here. And now the actual splitting apart of the two chromosomes occurs as the cell elongates. The cell undergoes a period of elongation where the two chromosomes are physically pulled apart from one another, and then the cell undergoes cytokinesis behind that, and divides them. There is one similarity to the plant cells, and that is most prokaryotes have a cell wall. But the cell wall does not develop across a cell plate. It develops on the edges here to pinch off the two new cells. In addition, just to remind you, most prokaryotes, in addition to this big chromosome, they also have small genetic elements, also in circles, little circlets of DNA called the plasmids. And the plasmids will actually replicate as well. Some plasmids are independent and just replicate on their own. Other plasmids are timed to replicate together with the chromosome. It just depends on the plasmid and often on their origin, because plasmids are often acquired from outside. So it just depends on the type of plasmid as to when they replicate, but they do it in exactly the same way. Okay, so we need to ask the question. We've said that we've seen all these different phases, interphase and interphase being divided in gap 1, S, and gap 2, and then this very rapid mitotic phase. These events are very carefully controlled by the cell. They are very carefully managed, and most of the management appears to be internal to the cell itself. There seem to be complex mechanisms whereby the cell is able to monitor its own status and move from one phase to another in an orderly fashion only when the correct metabolic conditions exist. This is very important because if, for instance, we can experimentally force cells into growth phases which are inappropriate. We can take a cell which is just divided and we can experimentally force it to immediately go into mitosis again. It's very, very dangerous, and usually it's fatal for the cell. So the cell needs to have a length of time during which it builds up all of its reserves, all the correct enzymes, etc., etc., etc. The frequency with which a cell divides depends very much on the nature of the cell. And so, for example, our nerve cells very seldom divide. They can divide, and they do, but they do so very, very seldom. Whereas, for example, our skin cells, the epidermal cells, they reproduce very, very frequently. Our mucous membrane cells reproduce even more rapidly. Their division is continuous. But each of these lifespans is determined genetically. The cell is, in effect, monitoring its own status very carefully. There is an exception to this, and that is when cells actually lose control. They lose control over their division status. And this is essentially almost a definition of a cancer cell. Cancer cells escape the usual controls of the cell cycle, and they are able to continually divide rapidly, grow extremely rapidly usually, and escape all of the normal control mechanisms. And that is what makes them so dangerous. That they, as we'll see in a while, we'll discuss it in more detail in a minute. Just remember, most cancer cells have escaped all of the control mechanisms that I'm going to mention to you. So the cell cycle that we saw, interphase G1, S, G2, and then the mitosis, the major driving factor for these, major control factors, are due to signaling molecules which are generated inside the cytoplasm. And I'm going to just mention one set of experiments which showed that this is true. So, for example, we can monitor cells maybe that we have in culture or whatever. We can monitor them, and we can tell very carefully where in their growth cycle they are. So what these workers did was they took cells, for example, which were in S phase, were busy replicating their DNA. And then they took another cell which was in G1 phase. It was just in its gap phase. It was busy growing, etc., after cell division. And what they did was you can actually get these cells to fuse. You can get those cell membranes, the plasma membranes around the outside, to actually blend with one another and fuse the cells. And in that case, the cytoplasm becomes united. And what they found was that they could immediately force this cell which is in G1 phase, to begin replicating its DNA inappropriately. It's not really to do it, but you can force the cell to do it. You haven't done anything else other than cause the cytoplasm to merge, and whatever signals were present in the cytoplasm here, which were maintaining the cell in S phase, now come to exert an influence over this cell as well and force it into S phase inappropriately. The second experiment was even more radical because they took a cell which was in mitosis, engaged in mitosis, and merged it to a cell which was in G1 phase. It hadn't even replicated its DNA yet. It replicates its DNA in S phase, it's not in G1. So here we've got a cell with unreplicated DNA. And now if we merge them, it forces the cell to begin mitosis, but it can't. It can't proceed with mitosis. It will begin, but it'll break down. The mitosis will be chaotic because there aren't duplicate chromatids to move apart. Nonetheless, you can see it start. You can see it building a spindle. You can see the nuclear membrane break down. You can see the chromosomes shortening, even though they're not duplicate chromatids, etc. A very clear demonstration that the major signals are from the are in the cytoplasm. This is not to say that these signals are entirely generated inside the cytoplasm. These signaling molecules in the cytoplasm that are circulating and telling the cell where to go, how to behave, may also be in response to events outside the cell. They may be generated due to internal events, but they may also be generated in response to external events, as I'll mention in a minute. There is very definitely a system which operates as a sort of molecular clock, which begins ticking immediately mitosis is finished. There is a molecular clock that runs and informs the cell about how long it has been in a particular phase. I'm not going to discuss it in the one mechanism in a lot of detail, and I'm setting it instead as an extra credit assignment for you. You'll see in your PowerPoints. So this cell cycle control system, as I say, is responding to events inside the cell, but it is also responding to events outside the cell, to signals from inside and to signals from outside. And the clock has very specific checkpoints where the cell effectively does a check, and if events are not exactly right, it won't proceed beyond that checkpoint. It's like a traffic signal for the cell, whether it can stop or go past this checkpoint. And there are three where it won't surprise you. There are checkpoints at G1, at G2, and in the M phase. And I'll just, for most cells, it's this G1 that is really important. So here's the G1 checkpoint. So this is, after mitosis, of course, the two juveniles, the two daughter cells are smaller. And it's during G1, they start to grow. They start to accumulate biomass and build up all the reserves and everything else that they need to do. This G1 checkpoint is one of the reasons that some cells very seldom divide. For example, nerve cells will go through an original mitosis that is their origin, but the cells grow to G1, to the G1 checkpoint, and they won't go past G1. They are genetically programmed to stop at G1, and they go into what we call a G0 phase, or G0 or G0. And that is an extended period with no replication of the DNA and with no cell division. And that would be typical of nerve cells, for example. They are in an extended G0 phase, and they very, very seldom divide. They can divide. They will, under certain circumstances, pass G1 and go on to divide, but it is not very frequent. Most other cells pass G1 once they have accumulated enough material, etc., to fund the rest of their phases. The second checkpoint is here at G2. This is the G2 checkpoint. It checks to see that, first of all of the synthesis has taken place, that the centrosomes are duplicated, for example, that the DNA has been correctly replicated, etc., etc., and that all the enzymes and everything necessary to shut down cell function during mitosis are present. And if it fails, cell division will not proceed. Cell division will not proceed until all of those conditions are met. Then there's a very important checkpoint here, and this is called this is the mitotic checkpoint. And the mitotic checkpoint occurs at metaphase, and it actually checks to see that all of the chromosomes are lined up perfectly on the metaphase plate. And it then checks to see that at anaphase, it checks to see that they have been correctly separated. And properly, if something has gone wrong, if like two chromatids have gone to the same side instead of to opposite sides, the cell mitosis will actually stop and the cell will be killed. It's killed by internal mechanism called apoptosis. That is an imperfect mechanism. We do see cells that have errors of division, but properly, for most cases, these errors result in the death of the cell. So that is the mitotic checkpoints. They check metaphase and then anaphase separation. Okay, once division is completed, then after telophase, the cell begins growing again, growing until it reaches G1 checkpoint, and we go through the whole thing again. The length of this cycle is genetically determined by the nature of the cell. As I told you, nerve cells very seldom divide. Things like skin cells, mucous membrane cells, very frequently divide and go through this very rapidly. Okay, so this basically just gives you the different checkpoints. Okay, so here is this is the little extra credit that I want you to do. I will post this to Canvas as well and post it as an announcement for you. 1.5%, 1 to 2 page discussion. What is the nature of the molecular clock in the cell? It's really fascinating and it's well explained in your book, okay? But I'm going to leave it up to you to actually handle. Okay, I want to just discuss with you a couple of things. I've tried to make it clear that in cell division, the cell is definitely responding to a lot of internal cues, to its own clock, its own status for growth, etc., etc. But I do need to tell you also that the cell is very much influenced by environmental conditions as well. And here are just a couple of them. There are many of them, but here are just a couple. A lot of these were discovered in the course of developing techniques of tissue culture, of taking living cells and putting them into artificial culture outside of a body and actually growing them and seeing what it was that actually allowed them to grow. This was a very difficult undertaking initially. Initially, when these attempts were made to culture cells, they were often unsuccessful. A lot of the really early, really successful tissue cultures were actually done with cancer cells, because cancer cells grow willy-nilly. They just they lack most of the control mechanisms that apply to normal cells, and so cancer cells are actually relatively easy to grow in culture for reasons that I'll explain as we go through here. So a very common tissue culture of animal cells is of fibroblasts. That is, those are skin cells. They're useful because they have a very short cell cycle, so they rapidly grow. And what they would do, they take a sample and cut it up into small pieces, and then enzymatically digest all the cell connections. So you ended up with 3 cells in your solution, and then they would be transferred to culture vessels. These culture vessels are usually these little flat flasks like this, and that is for a very important reason that I'll show you in a minute. One of the very first things they discovered was that these cells, in order to grow, in order to proceed through the cell cycle, they required the presence of substances called growth factors. There are various growth factors, but one of the most important is this one, PDGF, that is platelet-derived growth factor. And platelet-derived growth factor appears to be essential for the growth of these cells in culture. So platelet-derived growth factor is a growth factor which is produced by the platelets. The platelets are very, very small cells in the blood, closely related to red blood corpuscles, but very much smaller. And their principal responsibility in the body is actually to promote clotting of the blood. But they also produce these growth factors that influence the growth of cells from outside. The growth factor is perceived by the cell. The growth factor becomes bound to a receptor. And once it's bound to that receptor, the receptor sends a signal into the cell to say that growth can continue, and that the cell can move through the cell cycle and to the various phases towards cell division. Without the growth factor, the cell stays quiescent and doesn't grow. So that's one. The next is, these cells, if you have them just swirling around in liquid, they don't grow. They require attachment. They have to be attached to something. And this is called anchorage dependence, and it applies in the body as well. It applies. Cells need to be attached to something. Often, they need to be attached to, in culture, they need to be attached to a surface. That's the reason we use these little flat flasks like this, because it's got a nice big flat surface underneath where the cells can settle, and they will then begin growing. But in culture, these cells, when they contact one another, the cell cycle stops. They stop growing once the cells are in contact with one another. They form a single layer. And this we call density-dependent inhibition. So they have, first of all, anchorage dependence. They need to be settled on a surface. But when they grow, they will stop when they become crowded. This is density-dependent inhibition. In the body, it's a little bit different. These same rules apply, but it depends what tissue you're talking about as to how the cells actually behave in relationship to one another. So remember that the tissue culture may be a little bit different to what happens in the body itself. One thing is clear, first, and that is the behavior of cancer cells. First of all, cancer cells often do not require any growth factor. in order to carry on growing. Second of all, cancer cells will continue to grow in suspension, but they do grow much better. They do have a certain amount of anchorage dependence. They will grow in suspension, but when they grow on the surface, they will very rapidly overgrow one another. They do not have this density-dependent control over growth. So it looks like our cancer cells are evading all of the control mechanisms. And that is exactly right. That is exactly how they behave. They are independent of many of the growth factors and things like that, either because they produce their own growth factor, which some of them do, or because their signaling mechanism, they have, remember I told you, the growth factor binds to a receptor that signals the cell. Those receptors are continually on. They're switched on the whole time. They're telling the cancer cell that the growth factors are present when they aren't. And the cancer cell just carries on growing. In addition, the cancer cells may evade those checkpoints during division of the cell. And in particular, they may evade the mitotic control, the mitotic checkpoints. Cancer cells very often in their division, they are inaccurate in their division. And that makes them even more dangerous because they become, the cancer itself comes to consist of different kinds of genomes, different mutations arise, and the central control, which is the control exerted by the nucleus over the functioning of the cell, becomes distorted. So cancer cells have many ways to evade the things that make a normal cell normal. I think this covers everything. Yes, it does. So one of the remarkable things about cancer cells, and something that made them very useful as an experimental tool, and still does, is that cancer cells are immortal. Most cells have a very defined lifespan. And at the end of that lifespan, the cell dies. An example would be a skin cell. Skin cells are produced continuously by a layer of generative cells that undergo mitosis. The generative cells stay, they continually divide, but they produce skin cells which layer up above them. And those skin cells have a lifespan of only days or weeks before they die. And they're just programmed into them. Nerve cells, very, very, very long-lived. So you may still have nerve cells that you had as an infant, for example. But it would eventually die anyway. Cancer cells are immortal. We have one of the earliest lines of cell culture was a culture of cancer cells derived from a lady called Helena Lax. And those are called HeLa cells, H-E-L-A cells, and they are immortal. She died in the 1950s, and her cells are still maintained in laboratories all over the world. And they are immortal because they've lost the timing mechanism. They have lost the ability of the cell to gauge its own age. They are immortal. We call cells like that, we say that they have undergone transformation. And transformation can happen for various reasons. It can happen because of a mutagen, chemical or other. It can happen because of a virus, or it can happen naturally during cell division. But somewhere along the line, they lose this essential requirement that cells have a limited lifespan and instead become immortal. We have a whole arm of our immune system, which is dedicated to looking for cells like that and killing them. But unfortunately, it does occur that some cells which have become transformed in this way evade the immune system and end up forming essentially a clone of themselves. The cells begin to divide rapidly in a set place. The cells lack the normal cell control. They lack the normal developmental controls. And instead of being part of a natural tissue, they instead begin to form a mass, which we refer to as a tumor. So we recognize different kinds of tumors. First of all, benign tumors have the characteristic that they usually remain exactly where they originated. And second of all, benign tumors usually have a covering around them of connective tissue that develops. Benign is a bad name for them. They are very often, a benign tumor does not cause enormous amount of problems, but it very much depends on where they are. So, for example, most brain tumors, which are extremely dangerous, are benign tumors. The opposite of benign is malignant. And the difference is that benign tumors, the cells of a benign tumor, remain associated with one another. They may, in fact, have quite strong ties to one another to keep them tightly bound. Also, they usually have a connective tissue around them. But a malignant tumor does not. A malignant tumor, the cells are far looser. They're loosely attached to one another, and they may break off, and they may move either through blood vessels or through the lymphatic system. They may move to another site, and there they act as seeds, and they may cause secondary tumors or metastatic tumors. So malignant tumors have this terrible property of metastasis, that they spread from an original primary site to secondary sites. Cancer cells have a couple of fallibilities, if you want to put it that way. First of all, they divide very, very rapidly. They often divide. And if we treat with anything if we treat with any method which damages DNA, especially during division, then we are likely to kill cancer cells more rapidly than any other cells. And this is the idea behind treating with high energy radiation, gamma radiation, whatever. If we irradiate an area with a tumor in it, we will damage cells which are engaged in cell division far more than cells which are not. So we'll tend to damage cancer cells more than others, but we may still damage other cells. Cancer cells also often lack repair mechanisms for their DNA, which normal cells have. So this is another reason we can treat with radiation, or we can treat with chemicals which damage DNA that is engaged in division especially. Normal cells can repair it to a certain extent. Cancer cells tend not to be able to. So cancer cells definitely have vulnerabilities. This is just an illustration of one of the more common cancers, and one which unfortunately still carries quite a high mortality rate, although treatments have improved enormously, and this is breast cancer. And breast cancer, again, begins as a small primary tumor, but depending on the type of tumor, not all breast cancers are the same. Several of the breast cancers are highly metastatic, and very early on, they begin to shed cells which go into the blood system or into the lymphatic system. And very often, these will become trapped in lymph nodes. Now, you may know somebody that has had breast cancer, that has had the tumor excised or breast removed, and also has had lymph ducts and lymph glands removed in the vicinity. And this is the reason why. The lymph glands actually act as a sort of a filter, if you want to think of it that way. They catch the metastatic cells and often the secondary tumors begin growing inside the lymphatic glands, the lymph glands. I think that I've just about covered everything. And so we'll leave mitosis there. And I am just going to actually start our next section, which is on meiosis, I'm sorry, but just give me 1 minute. Okay. All right. Mitosis. Mitosis under normal, we ended up talking about cancer cells where it's a little bit weird, but under normal circumstances, mitosis, a diploid cell inside the body divides and ends up producing 2 identical diploid cells. And that is because the DNA replicates. When we look at a normal chromosome, it consists of 2 chromatids, which are identical to one another. And during the process of mitosis, we split identical chromatids apart from one another. So that's one form of division, cell division. But there's another very important form of cell division, and this is the process of meiosis. In the life cycle of most eukaryotes consists of a lengthy period of time when the cells are diploid. For us, most of the cells of our bodies are diploid cells. We have 46 chromosomes in almost all of our cells. And those are the somatic cells. The cells that make up the body itself are the somatic cells. 46 chromosomes. So, you know, of course, that we reproduce by producing sperm and we produce eggs. And the new fetus is produced from fertilizing the egg. So you've got an egg and a sperm unite to form the fertilized egg. Well, how is that done? Because if the egg had 46 chromosomes, right? And the sperm had 46 chromosomes, and we put them together, we would end up with 92, right? Chromosomes. Okay, 92, right. So that organism goes off. It's got 92 chromosomes. And now it develops from mitosis. So all its cells have 92. What about when it wants to reproduce? So you've got 92, Mr. 92 meets Mrs. 92, and they reproduce. Now we've got 92 plus 92, 92 in the sperm, 92 in the egg, and now the fetus has what? 184, right? That's impossible. We can't do that. We would soon have, we would not be able to fit all the DNA into the cell if we carried on like that. No, something else has to happen. There has to be a mechanism whereby we halve the amount of DNA that's present in sperm, and we halve the amount that is present in the eggs. In fact, we have to halve the number of chromosomes present in the egg, halve the number of chromosomes that are in the sperm, and put them together. Now, human beings, we would have, instead of 46 chromosomes in the sperm, we have 23. And in the egg, we have 23 chromosomes. Now, sperm fertilizes egg. What do we end up with? 46 in the fertilized egg to proceed through mitosis and ensure development of the embryo. We have to have a mechanism to halve the number of chromosomes, to halve the amount of DNA. And our gametes, sperm and egg, are haploid. They only have 23 chromosomes. They do not have 46. And they have 23 individual chromosomes. They do not have paired chromosomes, the way we do in the diploid. That is the fundamental purpose of meiosis. But meiosis is an absolutely incredible process. And first of all, let's just have a look. We'll ask some questions. Here is a family, and the mother, father produced sperm, mother produced eggs, and they had 2 beautiful daughters. And the first thing to notice is that these daughters are extremely have a lot of similarities to one another. The hair, the color, just in general, they have a lot of similarities to one another. They also, if you look carefully, look at this lady and look at her father. Can you see similarities? There are lots of similarities between them. Why? Because the parents hand on information, they hand on genetic information to their progeny. The genetic information is handed on in their gametes, in the egg and in the sperm. So a child inherits genetic information. It ideally inherits all the necessary genetic information for the organism to exist and be healthy and grow and everything else. So the first thing we notice, parents and their children, is this huge similarity between them. But it's not the similarity that I'm so interested in, so much as the differences. First of all, this is the same father, this is the same mother. Here are two daughters. Yes, there are similarities, but look how different the two daughters are to one another. And look how, in fact, how different they are to the parents. There are similarities, but they're not identical. Why aren't the two daughters identical? It's the same father, same sperm. It's the same mother, it's the same eggs. Surely we would expect them to be far more similar. No, they're not. They're very, very different. And siblings can be extremely different to one another. Extremely different with the same parents. Here's a marvelous pair. These two girls are twins. They are fraternal twins. And the mother is a white lady with red hair. And the father is African American. And this is when they were little. Here they are grown up. These are fraternal twins. There is no doubt about their origin. They have the same mother, the same father, yet look how incredibly different they are. In talking about reproduction, we need to be able to explain these differences. And we can very, very clearly explain those differences if we understand the process of meiosis. The process of gamete formation explains this exactly, how this can occur. And this kind of variability produced through meiosis, through the process of sexual reproduction, is one of the most important processes in all biology, because this variability is so important in terms of evolution. And it is a driving force, and a lot of this variability arises because of the process of meiosis. So it behooves us then to actually understand a little bit about how meiosis works. So don't worry about the details of this diagram for the moment. We will get there, but I just want to point out right at the very beginning, I want to have it clear to you what some of the differences are between mitosis and meiosis. First of all, mitosis produces two genetically identical progeny, right? That you start off with a parent cell, and that parent cell divides, and you end up with two daughter cells, which are identical to one another, both diploid, both with exactly the same DNA, exactly the same DNA sequence present in their nuclei. Two cells. In meiosis, we're going to see that we end up producing 4 cells. We produce 4 cells, and they are not genetically identical. They are not necessarily, in the chances of them being genetically identical are extremely remote, as you will learn. So in mitosis, what we did was we took our chromosome and we duplicated it exactly. right? We had two chromatids that were identical to one another. Then during division, we split them apart. So we had identical DNA here, identical DNA there. Now in meiosis, we're going to see that in fact, we go through two kinds of division, two division cycles. And in the first division cycle, we actually take whole chromosomes and move them apart. We start with 46 chromosomes in a cell. And in the first stage of meiosis, we'll move whole chromosomes apart from one another, and we'll end with 23 on one side and 23 on the other side. There, bingo, right at the very beginning of the process, we've halved the amount of DNA. We've halved the number of chromosomes. But all sorts of other things happen during meiosis as well that we need to talk about. And so here's just taking this as a simple example to emphasize it. Here's this has two chromosomes in a parent cell, and these chromosomes actually are derived from the parental. They derived one from the mother and one from the father originally. Although this is the father, this example. During replication, each chromosome is replicated. Those are the two identical chromatids, homologous chromosome, homologous chromosome. In meiosis, what we would do is we would split these chromatids apart. But in meiosis, what we do is we split whole chromosomes apart. We take that chromosome that side, we take this chromosome that side. And we've ended up now, immediately, we've ended up with half the number of chromosomes. There's only one chromosome. There are two here. There's one there and one there. And now in meiosis, we undergo a second round of division. It looks just like mitosis. But now we split this chromatid away from this chromatid, there. We split this chromatid there and this chromatid there. What we've ended up with from one cell, we ended up with 4 cells. And you will see very clearly in a while why it is that we say these 4 chromatids are not identical to one another. They differ from one another. We end up with 4 unique cells. One of these, this is the father. Let's say this one goes to form a sperm. This one here goes to form an egg. This is the mother's side here. And they unite in the act of fertilization. And we end up, bingo, we end up back being diploid. These here, those there, and these here are haploid. They have half the number of chromosomes. They only have, in humans, we look at the whole cell, they would have 23. These are the gametes. These here, we refer, the parent cell that's going to produce these, the gametes, is called a germ cell. And it has the unique ability, unique inside the human body, that it can produce cells which will become haploid. The only haploid cells in our bodies are those which are destined to become gametes. The rest of our cells are diploid. Take the two haploid gametes, put them together, and we end up with a diploid fertilized egg, a diploid zygote. The zygote is the result of fertilization. And this now proceeds, just let me do one thing, sorry. This now proceeds through mitosis, through the process of development, proceeds through mitosis to produce the embryo. Okay, so some very, very, very important things are happening in meiosis that we need to understand, because we have to understand them if we want to understand the process of inheritance, the process of heredity. And especially, we have to be able to explain how it is that even though we have one mother and one father, father producing sperm, mother producing eggs, siblings are by no means identical, unless they are identical twins, which is a different situation. Identical twins arise from one fertilization event. But siblings which arise from independent fertilization events differ from one another. And if we look at the whole range of the human population, you would have to wait many billions of years before one person was born who was identical to another person who was born. We can say that with mathematical certainty. And we need to be able to understand why. And so, first of all, if we follow the transmission of genes during heredity, then that we are investigating genetics. We are investigating how it is that offspring inherit characteristics that the parents have. But we also need to bear in mind that there is this enormous amount of variability as well. And much of that variability is going to, we see, is going to be produced because genes don't operate independently of one another. They interact with one another. And the process of meiosis actually is a process of mixing and matching genes from the mother and the father in the sibling, in the offspring. And different offspring have a different mix and different match of genes. And that mixing and matching takes place during meiosis. So let's just remind us, this is just, you need to remember all of this taxonomy of what we're going to be talking about. Genes, they're the unit of heredity. And they are made up of coherent segments of DNA. And in general, it's not entirely true, but in general, you can think one gene equals one polypeptide. Because genes, by and large, are the recipe for polypeptides, which will go on to form the functional proteins of the body. And those genes are passed via gametes to the succeeding generation. And the gametes are our only haploid cells. And they, in humans, sperm and egg are each haploid. When they unite in fertilization, then the ploidy is restored, and the zygote is diploid, formed from the union of two haploid cells. All of the cells of our body are called the somatic cells, and those which are involved with gamete production are called the germ cells. So I'm not going to worry about talking about asexual. Remember that there are many organisms that reproduce quite happily asexually, but they're essentially producing a clone of themselves. They're producing genetically identical versions of themselves. Sexual reproduction is extremely specialized, and it is highly, in terms of biology, in terms of ecology, in terms of evolution, it is highly, highly advantageous because sexual reproduction produces such a range of variability in the offspring. And that variability is because each of the offspring inherits new combinations of genes from the parents. They are born of egg produced by the same mother, sperm produced by the same father. But the siblings, if we were to examine the combinations of genes that they inherit, they differ in the different siblings, and that produces the variability that we see. We call it a life cycle. The life cycle for us, most of our life cycle is a diploid life cycle. Our only haploid cells are the gametes, and their lifespan on their own is quite brief. And as soon as they're fertilized, we produce a diploid zygote. And if we were to look at we can actually experimentally do a preparation where we get human cells induce them to divide, and right at usually at metaphase, somewhere around there, we experimentally extract the nucleus, and we can effectively lay it out and stain the chromosomes. Remember, the metaphase, now the chromosomes are all beautifully thickened and wound up, so they're easily visible. This is now done by computer. It used to be done, you take a photograph and you cut out all of the chromosomes. When we do that, for somatic cells of the human, we find that there are 23 pairs of chromosomes. And the chromosomes can be matched one to the other in pairs, in what we call the homologous pairs. One derived from the father, one chromosome derived from the mother for each of the pairs. They are homologous. First of all, they look like one another. That's how we can sort them. They stay in the same, and they're the same shape, and the centromere is in the same place, etc. So we can match them quite easily. If we were to examine the genes, we'd find that the same genes are present on the chromosome, on each of the chromosomes. These chromosomes here, in actual fact, have already duplicated. You just can't see the two chromatids. But here's a diagram here. You can see they're closely stuck to one another. There are special proteins that bind them closely to one another. So this is one chromosome there. There is its homologous chromosome. They have the same genes at the same position. So for example, if we were looking, maybe this one has a gene for eye color there. This one, the homologous, it'll have a gene for eye color as well. It just may have a gene for a different eye color. So when we look here, we see here's this chromatid here is identical to that one there in every respect. The DNA sequence is exactly the same. This chromosome here is very similar to this one. It has exactly the same genes, but the genes may differ. The DNA sequence may differ between this chromosome and this one here, but they are very, very similar. They are homologous. These are called the sister chromatids. These are the homologous chromosomes, one from the father, one from the mother. Go through the whole human genome, lay them out like this, and we call that a karyotype. So if we look at the human karyotype, we see 23 pairs of chromosomes. Most of them, the chromosomes in the homologous pairs, are very, very similar to one another. You go all the way through, except for pair 23. There's pair 23 there. In females, the pair 23 look very similar to one another. But in males, one of the homologous pairs, is this short, stubby little chromosome here. And this we call a Y chromosome. And the Y chromosome is the chromosome which determines maleness. It actually has genes on it which determine if these genes are present in a zygote, the fate of that zygote is to develop as biologically male, if it has the Y chromosome that is fully functioning. If, on the other hand, the two of these are present, then the zygote will develop as a female. And that is determined, whether you're male or female, is determined by the nature of the sperm that fertilizes the egg, because the sperm can only carry either X, this chromosome here, or Y, this chromosome here. If the egg is fertilized by a sperm with an X chromosome, the zygote will develop as biologically male. If it is fertilized by sperm with an X chromosome, it will develop as biologically female. So that's pair 23. They're called the sex chromosomes. All the rest are called the autosomes. So all of these are the autosomes. The one pair that you might already have heard of is this one here, pair 21, because very rarely something goes wrong during gametes production, and a sperm or an egg may have 2 copies of 21 and then acquire another copy in fertilization, and you end up with 3 21s, and that is trisomy 21, also called Down syndrome. And many of you, I'm sure, know people with Down syndrome. They have 3 chromosomes at 3 chromosome 21s. So the number of chromosomes can have very profound effects. The nature of the chromosomes can have profound effects. If you inherit an X, you develop as biologically male, all things being equal. 3 21s, Down syndrome. So we're watching very profound things happening here, and we need to start to understand. Our first move towards understanding genetics is that we must understand meiosis. If we understand meiosis, you can understand a lot about the genetics that we're going to talk about a little bit later. Okay, so to labor the point and to really get this clear in your heads, because if you can comprehend and remember this now, it'll make it much easier for you as we start to discuss the mechanisms of meiosis. This little diagram here, we're looking at an organism with 3 paternal and 3 maternal chromosomes. So there are 6 chromosomes altogether, and that is 3 pairs. Here are the homologous pairs. There's the one, there's the homologous pair there. Here's the second, homologous, homologous. Those chromosomes are homologous. These are homologous. Each of the chromosomes consists of sister chromatids, closely applied here to one another. And the homologous chromosomes, very similar, but not necessarily identical to one another. The chromatids, necessarily identical to one another. And in this case, the diploid number is 6. And that the diploid, we use this notation, 2n. So 2n diploid is 6. And in the process of meiosis, what we're going to do is we're going to halve the number of chromosomes that are present in a cell. And thus we're going to produce a haploid cell. In the humans, the diploid condition is 46. The haploid condition is 23. And if at the end of fertilization, we take 23 from the egg, 23 from the sperm, we end up with the diploid condition again, which is 46. Remembering that if there's 2 X chromosomes, it's a female, biologically female. And if it's a Y chromosome, then it's biologically male. So a sperm cell can carry either X or Y, cannot carry both. This is our life cycle. We spend most of our time is spent in the diploid condition, of course. Haploid condition only for production of eggs and sperm. Fertilization recreates the diploid zygote. 2n, 46. I think it's just biosynthesis. This is true for most animals. With it, the majority of the lifespan is spent as a diploid. It differs sometimes in plants and in some primitive things, but for all our purposes, we can consider most of our lifespan as being the diploid, and our only haploid period is the formation of the gametes. So the process, I'm going to just introduce the process of meiosis, and then we will continue this next time. We'll complete this next time. First of all, meiosis is similar to mitosis in that the cells which are going to go on to go through meiosis still undergo the period of synthesis. They still undergo the period of replication of the DNA. So that the chromosomes which enter meiosis are in that typical form that we saw already, where there are two identical chromatids joined together. And in each cell, it's diploid, so it has one maternal chromosome, one paternal homologous chromosome. Meiosis, into the process of meiosis, you undergo 2 rounds of division. And you're going to undergo 2 rounds of division with no replication of the DNA again. Replication of DNA precedes all the divisions. At the end of meiosis, we're going to see that there are going to be 4 progeny cells. There are 4 cells, and each cell is going to have half the number of chromosomes. They're going to have the haploid number of chromosomes. So here's basically the process. First of all, here are the homologous chromosomes. This would be in G1 phase of interphase. Here they are in S phase, synthesis phase. And here's the homologous chromosome, homologous paternal, homologous maternal. Here are the 2 chromatids, paternal, the 2 identical chromatids, maternal. Here's the critical stage, meiosis 1. That's the first round of division in meiosis. All sorts of magical things happen during the process of meiosis 1. But for the moment, all that you need to get clear in your head, look what happens. Meiosis 1, we split this chromosome, the paternal, away from the maternal. We take the paternal there, and there goes the maternal. What happened in mitosis? How is division different in mitosis? In mitosis, we split chromatids apart, right? If I split chromatids apart to each cell, I end up with the same number of chromosomes, exactly the same DNA sequence present in each of the cells on either side. In meiosis, I'm doing something completely different. In meiosis, I'm taking entire chromosomes one way, and the other, the homologous, in this instance, just the one, it's going this way. What you need to remember is we have 46. In this cell, we have 46. 23 are going this way, 23 are going that way. Not only that, it's random as to which way they go. So the 23 on this side are a mixture of paternal and maternal. The 23 on that side, it's a mixture of paternal and maternal. And we can't tell which way they're going to go. It's a random process. The only rule is you can only have one of each homologous pair in this side and one of each homologous pair on that side. So immediately, meiosis, at this stage of division, we call this meiosis 1. In meiosis 1 in the human being, we would end up with 23 pairs on that side, 23 pairs on that side. We have halved the amount of DNA. We have halved the number of chromosomes. Now we proceed through another set of divisions. And this time, the divisions are very like mitosis, because now we're going to split this chromatid away from that chromatid, there and there. We're going to split this chromatid away from that chromatid, there. And we end up with 1, 2, 3, 4 cells. 4 cells, all haploid, all N. In the case of human beings, it would be 23. Each of these would have 23 chromosomes. Okay, so there's a lot more that we need to know about meiosis, but we'll leave it till next time to complete this.